Welcome back, everyone. I now invite Professor Yuichiro Takahashi to give his talk.
Okay, thank you very much. Yesterday I talked about lecture 2, but I couldn't finish everything, so just starting in the beginning of the rest of my talk, and then move to lecture 3. So this is just to remind you of chlamydomonas cells. So mainly I will present you some our data using the chlamydomonas as a model organisms. Okay, maybe so this is I talked yesterday sexual mating. There's two types, mating types, plus and minus, and if you cross with or mating type plus and mating type minus, so chloroplast DNA is inherited from the mating type plus cells, although the mitochondrial DNA is inherited from mating type minus. So that's what we discussed. So, for example, this is the example I talk. Mating type, if you cross, genetic crossing of mating type plus and minus, and this is chloroplast. So gray is antibiotic sensitive, resistant, and this is sensitive. And then if you cross the young zygote and the zygospore, and then germination, then 4 daughter cells appears. Okay, so basically the chloroplast, the DNA in mating type plus is inherited for daughter cells, and the nuclear genome is inherited in a Mendelian log 222. Okay, this is tetrad. Okay, so this is the typical style. So chlamydomonas is the very in the beginning it was used to as a model organism because of this genetic crosses is possible. So you can cross 2 different strains to see what happened. So that's and also chlamydomonas has ability to grow heterotrophically in the presence of carbon source like acetate. So you can make the mutant deficient in photosynthesis. Okay, that is very important for the studying photosynthesis. You can make many photosynthesis mutants. Okay. So there's actually plenty of collections of the mutant deficient in photosynthesis. So we can usually use this mutant to analyze photosynthesis. So that is one of the very important advantage using chlamydomonas as a model organism. Okay, so now this is a Kansai Osaka airport. I just departed a few days ago to come here. So we have Japan has exposition Expo 2025. Maybe I don't know, one month left. So Japanese pavilion present in this Expo 2025 about importance or usefulness of algae. No, algae can grow very well in and also produce many variable products or fix carbon oxide to reduce carbon oxide concentration in the atmosphere, or very environmentally, this is very useful algae. Not only Chlamydomonas, green algae or other brown algae or red algae is very useful. So Japanese pavilion appearing this, and they show some character of this Chlamydomonas. It's maybe misleading that Chlamydomonas look like animal, but actually this was made from designer crossing algae and Hello Kitty, that's cat. Okay, so this is genetic crossing between Chlamydomonas, you see, and also kitty. Of course, this is impossible, practically, right? So this Hello Kitty is evolved from the Miffy. Okay, the rabbit. So it's taken from the idea, but commercially successful kitties. Okay, this is just the end of lecture 02, and I move to lecture 3. So biogenesis LHC, light harvesting complex, and expression important membrane insertion and assembly with chrome 4. So in this limited time, I mainly focus on this import membrane insertion. So this is just an example of the. I told you yesterday about some plants grown in the dark becomes etiolated. Okay, so this is the example of the Chinese chive. This is popular vegetable in our country. So this is a green one is cheaper and more nutrition, good for young people, right? And if you grow this Chinese chive covered from the sunlight, it becomes yellow chive, Chinese chive. So this is very texture is very delicate. Taste is somehow different and a bit more expensive. Like green asparagus and also white asparagus, right? So yellow one is for probably elder people with more money. Okay, so this is HO-rated. So if you look at the plastid of this green and yellow plants, the green plant has a chloroplast, as you know, and yellow plant has HO-plast instead of chloroplast. So inside the membrane system is a little bit some crystalline membrane structure like this that is called prolamellar body. And some the membrane is extended, but it's very short and small amount. So then if you eliminate this, it contain does not HO+ do not contain any pigments. I mean chlorophylls. Some pigments, protochlorophyllite, a precursor of chlorophyll, accumulate and some carotenoid. So it looks a little bit yellowish. It's not green. So the reason why when the plant are grown under the dark condition, why it becomes etiolated is one of the steps of the chlorophyll biosynthesis is light dependent. So protochlorophyllite is converted to chlorophyllite by protochlorophyllite oxidoreductase. And in most the land plants, high plants, vascular plants, which becomes yellow in the dark, have only LPOR, light-dependent protochlorophyll oxidoreductase. So this enzyme is active under the light. So if you grow, this activity is deficient. So this conversion of protochlorophyllite to chlorophyllite stops. So chlorophylls do not accumulate. Okay, but in the green algae, Chlamydomonas, and some needle plants, they are green if you grow in the dark, because this organism contain dark operative protochlorophyllite oxidoreductase. So it's still this contain both light dependent and dark operative oxidoreductase. So even in the dark, for example, Chlamydomonas still becomes green and synthesize the chloroplast and green and thylakoid membrane. Okay. So this is an enzyme. Dark operative oxidoreductase uses ATP and NADP electron to this double bond is reduced hydration. So this uses ATP, and this uses light and NADPH as a reductant. Okay, so this part is just electron is coming. So that's a difference. But in a Chlamydomonas, it's green if grow in the dark, but the mutant that is called Y1 mutant in Chlamydomonas is deficient in this dark operative oxidoreductase. So that's what happens. So that is called Y1, yellow in the dark mutant 1. So this does not synthesize chlorophylls and becomes yellow in the dark. Okay. So start to synthesize chlorophylls and photosynthetic apparatus upon illumination. It's like vascular plants most. Okay. So if we grow the cells in the dark, it becomes yellow. The chloroplast becomes etiolated, and then during illumination, it turns to green, yellow-green, full green. So protochlorophyllite, it becomes chlorophyllite in the dark, in the light dependent reductase, and then chlorophyll A and B are finally synthesized. So the cells grown in the dark, and then harvest the cells and extract the pigment from the cells with, for example, methanol, organic solvents, because the chlorophylls are very hydrophobic molecules. So you need to extract the pigments, chlorophylls, and carotenoid with organic solvents. So methanol is very good solvent to extract the chlorophyll directly from the cells. So the color is like this, the yellow, not completely yellow, but it's a slightly orange yellow. Okay, because the carotenoid, some carotenoid are yellow or some orange. Then you illuminate the cells from 1 to 10 hours. The color becomes green, like this. So about in this case, about 8 hours later, the cell becomes fully green. It's relatively rapid, I think. In the plant, it takes longer time. So if you in the laboratory, if you work very hard and long, you can finish your experiment in one day. You have to wait 8 hours, and also after 8 hours, you collect the cell and do something. So, but still possible to do in one day. So this is an absorption spectrum. You know, this is wavelength, and this is absorbance. So in the yellow cells, grown in the dark, extract the absorption spectrum. Extract is this orange. So this is a 3 peaks here in the blue region. Is typical absorption spectra shape of carotenoids. Okay, this means xanthophylls and the carotene, the mixture. And if you look at this red region, you don't see any absorption here because it lacks chlorophylls. The chlorophyll has absorption of this red region and blue region, but this obviously this lacks chlorophyll, A and B, both neither. Then during illumination, the absorption in blue region increases like this, and also red region increases like this. Okay. So chlorophyll also has absorption in blue region, so this increases also very much. But this is from 8 hours. So 10 hours a little bit more pigments because after the cells becomes fully green, it starts to divide. So it starts growing. So this is what happened in the cells. So this mutant is useful for analyze the synthesis or biogenesis of chlorophyll protein complexes because when it grows in the dark and no chlorophylls, that means the chlorophyll binding proteins do not accumulate. Okay. Then if you illuminate the cells or dark brown cells, they start to synthesize chlorophylls and also start to accumulate chlorophyll binding protein. So you can follow how things happen for chlorophyll binding proteins. Okay, so this is a schematic model of the thyroid membrane, and there's a huge multi-protein complexes, mainly 4 multi-protein complexes involved in electron transfer reaction. This is photosystem 2, and this is cytochrome B6F, and this is photosystem 1, and this is ATP synthase. Okay, so do you know of course you know the chlorophylls, where the chlorophylls are present in the cell. So it's specifically present in the chloroplast, and more specifically in the thyroid membranes, and even more specifically present in the chlorophyll binding proteins, not in the lipid, in the proteins. Because the chlorophylls are very hydrophobic proteins, so it cannot dissolve in the liquid aqueous phase. And of course, it's maybe possible to present in the cytochrome membrane by lipid bilayer, but that is non-dissociated chlorophyls. For example, if they present in the lipid bilayer, once this so-called free chlorophyls absorb the light energy, do you know what happens? The light energy absorbed by free chlorophyls cannot be used for photosynthesis, so that is very toxic. So this high energy state of the chlorophyll, excited state of chlorophyll, react with oxygen and produce a reactive oxygen species that's also damage to the photosynthesis apparatus and total cells. So that's very dangerous. So free chlorophylls should not be present, should not accumulate in the chloroplast. So the chlorophylls must be present in the proteins that is involved in photosynthesis. So you know that the photosystem 2 associate chlorophylls, photosystem 1 associate chlorophylls, and also light harvesting complexes associated with PS2 or associated with PS1. Also, this light harvesting complex associate many chlorophylls. And also, one chlorophyll molecule are present in cytochrome B6F complex. This is, I think, important. Also, one carotenoid is present in cytochrome B6F. But we don't know why cytochrome B6F contain chlorophyll A and one carotenoid. Because in the respiratory chain, counterpart of cytochrome B6F is cytochrome B1 complex, very similar, but in the respiratory chain, mitochondria, cytochrome B1 complex contain no chlorophylls, no carotenoid. Okay, so we still don't know why. Maybe structural stability is the presence of the chlorophylls and carotenoid may stabilize B6F complexes, but still it's a kind of still open question. Okay, so what happens when greening? That is greening. Yellow cells becomes green during the light illumination is we call greening. So what happened during a greening? So this is 2 hours. After 2 hours illumination, this is 10 hours, fully green. So what happens? So already we isolated membrane, cytochrome membrane that is was not present in HO-rated yellow cells, yellow chloroplast, but start to synthesize the cytochrome membrane during illumination. So 2 hours later, it's already the amount of cytochrome membrane is detectable. So we isolate, although it's a small amount, the cytochrome membrane from 2 hours illumination, after 2 hour illumination, and 10-hour illumination. Then the membrane was solubilized with mild detergent, and then separated this extract. Extract means solubilized cytochrome membrane on the sucrose gradient. Have you ever seen this? So this is the tube is usually stand. So this is the top of the gradient, and this is the bottom of the gradient. And then spin for overnight. So depending on so this is the top is 0.1 molar sucrose, and this is a 1.3 molar sucrose. So then larger complexes sediment faster than smaller complex. So you can separate the large complexes on this sucrose gradient ultracentrifugation. depending on the size, basically. Okay, so you can find a very dense green band here, and a weak band here, and a very slight band here from this. But from 10 hours, fully green one, you see a 3 major band and also that's the main change. And then you can see what? This is we named A1. This is light harvesting complexes. Okay. And this is a A2 band is photosystem 2 core complexes, and also photosystem 1 core complexes present. And here, A3 band is photosystem 1 core complex associating light harvesting complex 1. PS1, LHC1 supercomplex, we called. Because we fractionate this gradient, we put a small pinhole on the bottom of the tube, and then liquid, we collect the liquid, the fractionate. So fraction, this is fraction. Fraction 1 is bottom, and fraction 20 is the. And then separate the polypeptide by SDS polyacrylamide gel electrophoresis. We call SDS page. And then this is immunoblotting data. So we checked the presence of PS1 subunit, LHC1 subunit only. But actually, this so this is LHC2 mainly, and this is PS1 is here, and here in after 10 hours, this A2 fraction almost PS1 it disappeared, and then A3 PS1 appeared. This is super complex with LHC1. So this kind of things is we follow the stepwise assembly of PS1 complex doing the greening. So greening experiment is very useful for this large chlorophyll protein complex. It's how this complex is assembled or synthesized. Okay, so this is a kind of way of thinking of the. Okay, so I talk this data later in the other lecture on the PS1 assembly. So I skip this. So another interesting thing is this is not greening experiment. This is just normal wild type chlamydomonas we used, and also pulse and pulse trace labeling experiments. We're using a radioactive isotope S35. So usually sulfur is 32, so it's not radioactive, but S35 is radioactive, so you can detect this by contacting before the X-ray films. Now it's intensified screening. We can detect the signal. Why this is very useful for? I mean, pulse labeling means the protein containing a sulfur, it's like a methionine or cysteine. It's labeled with 35 for a short period. In this case, only 1 minute. Okay, so the cells are constantly synthesizing proteins, but you cannot detect, say, the difference between if you look at the proteins. This is already existing protein or just synthesized protein. You cannot distinguish, right? old protein and new proteins. But if you label the protein with radioisotope, you can detect only newly synthesized protein. Okay? So after 1 minute labeling, you can stop the labeling by adding non-radioactive sulfur, S32, or sometimes you can stop by protein synthesizing synthesis inhibitor. But usually we don't use this because Chase experiment is after pulse labeling, you stop the labeling by diluting radioisotope with non-radioactive sulfur, and then you follow the fate of the labeled protein. Okay, so that's its Chase. The Chase, the destiny of the newly synthesized protein. This is after the 1 minute pulse labeling, and we waited for 2 hours. Chase, and after 2 hours, labeled what happened to labeled proteins? This is the method too. Okay, so this is also after labeling, we isolated thyroid membrane, and also we solubilized the membrane and fractionated on sucrose sensitive gradient. Okay, so this is fraction from bottom to the top. Okay, and the same thing after pulse label and the Chase. So you see at a glance, you can see the difference of labeling pattern between here and here. Okay, then you can know what happened. So after 1 minute, you can strongly see a strong signal of about 33 kilodalton proteins. This is a very famous protein that is called D1 protein of photosystem 2. That is a PSBA genes of chloroplast genes. It encodes these proteins. So maybe I will talk later in other lectures that photosystem 2 is turning over rapidly during illumination because PS2 is very easily damaged in the light, and then damaged protein, particularly the D1 protein, this PSBA protein, damaged and then replaced by newly synthesized PSBA protein. So that's why this protein is so strongly labeled. Okay, and this is photosystem 1 reaction center protein, about 68 kilodalton, and also other small proteins. And here, about 30 to 25 weak signal you can, but it's sharp and clearly you can see. This is light harvesting proteins, light harvesting complex apoproteins. Okay. The after Chase experiment, then what happened? Do you remember that the fraction A1, A2, A3, this is mainly LHC2, LHC light harvesting complex separated. This is mainly PS2, and this is mainly PS1. But here, PS1 was not there. The newly synthesized PS1 temporarily stay in A2 fraction, smaller subcomplex. The PS2 is already here, but you can see a smaller fraction, the fraction that separate the smaller subcomplexes. You can see a still strong signal. So probably this is a not fully assembled PS2 protein. Okay, so after separating on sucrose centigrade, you can see how the newly synthesized proteins are distributed. Then after 2-hour chase experiment, or for example, photosystem 2, this part, smaller fraction, PS2, almost disappeared. Shifted to H fraction. So at least 2 hours, newly synthesized PS2 protein is completely assembled and separated this fraction. Okay, and also PS1 here, A2 fraction was more shifted to A3. And the PS1 other peripheral subunit also present here. But this PS1 is still here, but it's not. So assembly process is slower in PS1, but still change. You can see the change in 2 hours. And you can see a stronger signal of light harvesting complexes here. Okay? Somehow. So this is interesting why the LHC2 takes longer time to assemble in cytokine membrane. This is a speculation, but this LHC2 proteins is encoded by nuclear genes. So these genes are transcribed in the nucleus, and then the messenger RNA is moved to cytosol, and then ribosome in cytosol synthesize these apoproteins as a precursor. You see, there's a little bit single peptide is N-terminal, is a little bit additional peptide is present in N-terminal side of the LHC protein. That is we call precursor protein. But this is already mature protein. So, and then it's translocated to the chloroplast, and then across that stroma, and then reach to the cytokine membrane, and then integrated, inserted into the cytokine membrane. Then we can detect this. So it took longer time than these chloroplast encoded proteins directly translated and directly inserted into the cytokine membrane, but in the nuclear encoded proteins takes a little bit longer time to accommodate in the cytokine membrane. Probably this data suggests this process. Okay, so it's interesting to how to say from the data what you can speculate. This is very interesting part of the science. Okay. So then I'm going to focus on getting time is anyway. I have another lecture.
No, you please go ahead.
So I should go on. So there are several types of the LH light harvesting complex, LHC light harvesting complex. One is the mainly associated with P photosystem 2. The photosystem 2 core, we call core, and then LHC2. The light harvesting complex associated with PS2 is we name LHC2. Okay, so light energy is absorbed by LHC2 is this excitation energy is transmitted to PS2 core. So this light harvesting complex makes the antenna size to harvest and collect light energy larger, okay, much larger. But also at the same time, its light is also collected by LHC2, but if photosynthesis reaction is some part is the limited, so light energy is excess. So in that case, PS2 does not need any more the light energy. So then it's dumped as a heat. Otherwise, PS2 is damaged. Then you have to resynthesize everything. So it's dangerous. And also another light harvesting complex, LHC1, that is associated with PS1 core. Very similar structure, but it's a little different. So this also energy transfer from to PS1 core to make size of the antenna size larger. So it's not so clear for me that it's some excess energy is also probably dissipated as heat if the light is excess. But this part is this for PS1 is less clear than PS2. Okay, so as I talked before, the LHC proteins, apoprotein, no cofactor like chlorophyll A and B and carotenoid, has a N-terminal extension signal peptide, the precursor proteins, and then from cytosol into the translocated into chloroplast across the two layers of envelope membranes at the site of translocon. Okay, a TOC and TIC. The TOC is a translocon complex in the outer envelope membrane, so outside outer membrane. So TIC is translocon complex in the inner envelope membrane. So these two cooperate and then translocate this apoprotein of LHC, okay, into stroma. And then the protein peptide cleaves off this signal peptide, then apoprotein. It becomes mature size, okay, from precursor to mature size. But this blue part is hydrophobic region of this protein because light harvesting complex is embedded in the thyroid membrane, hydrophobic region. So this part is transmembrane helices are hydrophobic. So it's difficult to dissolve in stroma. No? To translocate across the stroma to the thyroid membrane, this must be becomes soluble. So two protein factors, CPSRP 43 and 54, assist this LHCP apoprotein remain to be solved in stroma. Otherwise, this hydrophobic protein may aggregate, between the hydrophobic region to make aggregate. That's very bad. So this is a chloroplast signal recognition particle. So it's associate this apoprotein and transport to thyroid membrane, and FTSY is a receptor proteins of this complex, and then ARB3 is inserted. So this ARB3 insert this apoprotein into the thyroid membrane. Then ALB comes from albino. So if this gene is knocked out, LHC cannot be integrated in the thyroid membrane, so LHC complex is not basically synthesized. So the more than half of the chlorophylls are associated with LHC, so the mutant cell becomes less green or very, how to say, the bleached color. So that's why it's called albino mutant. Okay, it depends on the organism. In the plant, it's completely white, and in the chlamydomonas, it's pale green. It becomes pale green. Okay, so I think I continue the next. So I think it's time for questions and discussion. Okay, I hope you understand the basic part, but I encourage you to ask any question or some part you don't understand. Just ask me. Yeah. Could you use microphone, please?
I have some question in sucrose density gradient.
Sorry?
Sucrose
sucrose
density
gradient.
gradient.
Which one? This one?
Not sir. Previous one.
Previous one? This one?
Yeah, sir. No, like in the LC complex are there in bottom, right?
Here, yes.
But in that Western blot, no.
I didn't use this in this experiment, the antibody LHC2. So this is obviously by staining. It's obviously we can see easily the LHC apoprotein. So this data was originally we used this data to follow the assembly of PS1. So we are not carefully checked, but it's obviously LHC2 proteins present here.
Okay.
Okay. Next.
Sensei, I have one doubt here. Yeah, here.
Okay.
Myself. So regarding the pulse and chase experiment, here particularly you are talking about the after pulsing. Regarding the chase experiment, the blots were there, no? So you are telling the LHCs have been formed more in the after 2 hours. So the intensity increases after stopping the labeling. That means how it possible the intensity will increase when you stop the pulse?
Okay. That's a good question. Of course, if you compare the PS1 and the PS2 labeling, it's not exactly the same, but it's comparable. But obviously, LHC proteins increases, right? So this is the protein in the isolated thyroid membrane, not whole cell proteins. So we are looking at the proteins, labeled protein integrated into the thyroid membrane. So if the protein is still in somewhere stroma or cytosol, we just removed during isolating thyroid membrane. That's why it should be the LHC2 apoprotein should be labeled in a similar way. But probably when we isolated the thyroid membrane, labeled apoprotein of light harvesting complex was still in a stroma or cytosol. So that's why that's my explanation. Probably that's true.
Thank you.
So it's important to how to how do you say, understand the raw data. Okay, usually if you look at the paper, the authors explain what happens. But I think it is also very important for you to look at the data carefully and then speculate what happened by yourself. Sometimes the author may explain wrongly, right? Explanation, interpretation may not be correct. But it's good for if you read the papers, you need to think about from the data what happened. Although the author describes explanation, but you try to think about by yourself to if the authors insist the right things or wrong. That is very important, I think. Other questions?
Question on TPU or LPU slide. TPU and LPU also.
Yeah. Which slide?
TPU or LPU.
LPR. DPR.
Yes.
This one?
My question is, both LPUR and DPUR reaction is necessary for the formation of chlorophyll ID. Means if a plant is illuminated or plant is going only in presence of light without any darkness, then can't it produce chlorophyll ID?
Cannot be. If this reaction is blocked or they didn't work, so subsequent reaction will not So in plant on those Chlamydomonas, this reaction is, in this case, Y1 mutant is blocked. So then protochlorophyllide accumulate. But it accumulates for certain level, but it stopped to, this does not accumulate enormously because there may be some feedback mechanism to block if the chloroplast accumulate this protochlorophyllide to certain amount, that suppress the upstream reaction. So this color is also represent this a little bit. Yellow color is probably xanthophyll carotenoid, and also this is I don't know exactly, but it's a little different. It's slightly orange yellow color. So that happens. So okay. So what is important is accumulation of the chlorophyll binding proteins and cofactor like chlorophylls and carotenoid synthesis, particularly chlorophyll synthesis and chlorophyll binding proteins synthesis must be coupled. If the chlorophyll is not synthesized, apoprotein becomes unstable without a cofactor, so it's degraded or somehow suppress the synthesis. And if the apoprotein is not synthesized and the chlorophyll is synthesized, then free chlorophyll accumulate, so that becomes toxic in the light. So this must be stopped. So these two reactions, apoprotein synthesis and chlorophyll synthesis, must be precisely coupled. Otherwise, it's dangerous to the cell. So, but we don't know exactly how this coupling occurs at the molecular level. We know the phenomena, but this is a very important question to be answered in near future. Okay, any?
Just I have a question. It's a long pending. I have one question. About the assembly. The light arresting complexes comes later, right? Comes later. Then
labeling. Okay.
Yeah, first labeling. But what are the determining factors like LHCP, right? LHCP and albino is very important. So the the cofactors for bringing all of them, even LHC 1, 2, 3, whatever. All of them will be the LHCP will be involved for all to bring from nuclear to the chloroplast. Oh, can you go to that slide?
The
Last one. Before one. Yeah, this one. The LHCP and FP.
sure, this is probably mean the complex.
Yeah, complex.
Yeah.
Okay, it's in the complex.
That's a good point.
But you know, we have LHCs, no, like LHC2B also there. LHC, I mean, PS2, LHC2,
sure.
and LHC1. So the mechanism will be the same for import all of them, all the LHCs.
Import.
Yeah.
Of this out.
No, other LHCs. Like, you know,
Other.
we have 9 LHCs, let's say chlamydomonas, 9 to 10 LHCs.
This mutant, for example, L03 mutant or SRP 4354 mutant has showed us very similar phenotype that lacks all the LHC. LHC1, LHC2, trimeric LHC2, and monomeric LHC2. These are all
No, this whatever you are showing is LHC, you know, LHC protein in the beginning, you know, precursor. It comes and enter into whatever, you know, to import to the thylakoids, right? The mechanism, or you know, the mechanism is the same for all other LHCs. Any other, like, you know,
probably very similar.
It changes to each subunit.
No, I don't think so. It's common. But this is only the nuclear encoded LHC protein.
Yeah.
But there are many, nearly 3,000 nuclear encoded protein in the chloroplast. But this system is only special for light harvesting complex, because light harvesting complex is the most abundant, one of the most abundant, but maybe next to the Rubisco large subunit. It's very abundant. So the most abundant membrane protein in this biosphere. So that's why probably the chloroplast developed a specific system to integration of only light harvesting complex.
I have one more question connecting to this. So which one will come? Is it a sequence of LHC 1, 2, 3, 4 like that? Or, you know, the sequence of importing inside? Will it be sequential, like 1, 2, 3, 4 of LHC 1, LHC 2, LHC 3? Which one will come first? And who determines this?
Actually, we don't have any data or evidence to distinguish this, but probably no. It's probably at the same time, because the assembly process is very rapid. So no, the cell chloroplast do not need to wait for sequential. It comes almost at the same time. So and then of course assembly order is not exactly the same, right? But we talk about other lecture, not here, but it's, once this apoproteins associate the pigments, that becomes stable. But without a pigment, apoprotein is very unstable. So the order of the assembly is there may be a order, but it's there's no regulation to synthesize this one as fast as second. But it's synthesized at the same time, but the assembly order, there's an order of the assembly. That's my speculation.
Just query.
Yeah.
That if there is any problem of LHCP, then you are telling that there are albino development, is it? So we generally see that albino kind of things in monocots in tissue culture. When you are going for a tissue culture using the hormones, we are getting a lot of albinos. So in that case, what is your opinion that? But sometime after some time, it become green. So what is the mechanism behind it? It is not exactly the mutation.
You mean the plant some albino, but it becomes under certain conditions?
Yeah.
It depends
particularly if you are going for a rice culture, in tissue culture.
It depends on the which the genes, the mutation, where the mutations.
No, it gives the albino color plant initially, then after it becomes green.
But in albino means not only this mutation, but other mutation also.
But if it is mutation, then in the next after 2-3 cycle, how it is become normal?
Okay, that's interesting phenotype, but
because this is a tissue culture general phenomena for rice. If you are going for rice tissue culture, you will be getting a lot of albinos. Even in the bamboo tissue culture, you will be getting a lot of albinos. But after sometimes it greens.
Oh, really?
Yeah.
Some genes are suppressed in the beginning?
We don't know.
Actually, in the Chlamydomonas, this ALK3 mutation and 4354 mutation, slightly light harvest complex is present.
Yes.
Slightly.
Okay.
I must say. But this complex is, if you analyze biochemically, somehow it's the apoprotein is very quickly degraded. So if you analyze polypeptide of this fraction of A1 fraction that is separated as LHC, apoprotein is the amount of apoprotein is very low compared to the amount of chlorophyll. So that means without this, there's another the route to assemble this apoprotein of LHC into the thyroglobulin membrane, but it's not efficient and it's not complete. So assembly process in the end may not work properly. So assembled or light harvesting complex, maybe somehow the structure is not like wild type. So it's probably degraded. But that is when we published the paper that the reviewer asked criticized. So we saw that there's another way, route, the minor route, but that is not cannot properly assembled. So this system is essential. Our conclusion is essential. Okay, so just